The 5-Year Journey of a Pitcher Plant. A Gardener's Time-Lapse Guide. Prologue. A Seed's Promise. The first time I saw a pitcher plant, it felt like I was looking at something plucked from a primordial world. It was a beautiful, alien thing, a leaf twisted into a carnivorous vessel, its rim a beguiling lure. My fascination grew into a quiet obsession, one that led me to a simple, audacious goal: to witness the entire life cycle of these plants firsthand. From the smallest seed to the full, magnificent flowering of maturity, my journey began with 2 packets of seeds. One for the tropical, mystifying Nepenthes and the other for the stoic, temperate Sarracenia. Little did I know that this simple act of sowing would unlock a decade of lessons in patience, adaptation, and the hidden miracles of the plant kingdom. The world of pitcher plants is divided into 2 distinct families, each occupying a different half of the globe and embodying a unique evolutionary story. First, there is the genus Nepenthes, a group of carnivorous plants often called tropical pitcher plants or monkey cups. This family consists of approximately 170 species, in addition to countless natural and cultivated hybrids. These plants are predominantly vining, with many species forming lianas that can climb several meters to reach sunlight. Their natural distribution is expansive, covering the Old World tropics from South China, Indonesia, Malaysia, and the Philippines, extending westward to Madagascar and the Seychelles, and southward to Australia and New Caledonia. The heart of their biodiversity lies on the islands of Borneo, Sumatra, and the Philippines, where numerous species are endemic to a single location, while many are found in hot, humid, lowland jungles. A significant number of species are montane, thriving in the unique conditions of warm days and cool, humid nights found at higher altitudes. In stark contrast, the American pitcher plants belong to the genus Sarracenia, a staple of the New World's carnivorous flora. These plants are herbaceous perennials that grow in clumps, their range spanning the Atlantic and Gulf coastal plains of North America. From the wetlands of Texas to the swamps of Florida and as far north as southern Canada. Unlike the tropical counterparts, Sarracenia species are adapted to a seasonal climate and a specific kind of habitat, permanently moist areas such as bogs, fens, marshes, and wet prairies. A variety of species exists, each with its own defining characteristics. The yellow pitcher plant, Sarracenia flava, is distinguished by its erect, trumpet-shaped pitchers that can reach 40 to 70 centimeters in height, with a yellowish-green coloration that may have a reddish-purple blotch at the opening. Its large, bright yellow flowers appear in early spring. The white-top pitcher plant, Sarracenia leucophila, grows to a similar height, but its tubular pitchers are green at the base, fading to a stunning white with striking red and green venation at the opening. The plant's name, derived from the Greek words leukos and phylon, literally translates to white leaf, an apt description of its distinctive coloration. This foundational knowledge of the 2 genera was the map for my horticultural journey, a guide to understanding the disparate natures and the unique challenges each would present. Year 1. From stratification to sprout. The first lesson in growing Sarracenia was a profound one in patience. The instructions for seed germination seemed counterintuitive for a living plant. Place the seeds in a damp medium and put them in a refrigerator for a number of weeks. I did as I was told, sealing the small container and tucking it into the crisper drawer. A bizarre act of horticultural faith. Months passed with no sign of life. While I waited, I learned that this process, known as cold stratification, was not an unusual trick for an impatient gardener. It was a fundamental biological requirement for these temperate plants. This need for a cold period is a key survival mechanism that has evolved over millions of years. Seeds from cold-hardy carnivorous plants, such as Sarracenia, produce a cocktail of enzymes that prevent them from germinating too soon. In their native habitat, seeds are typically produced in the early fall, just before the first frost. If these seeds were to germinate immediately, the tiny, fragile seedlings would not have enough time to establish themselves before the brutal conditions of winter set in, leading to their certain death. The cold, damp conditions of a typical winter deactivate these enzymes, signaling to the seed that the cold season has passed and it is safe to sprout when temperatures rise again in the spring. This intricate process is a perfect example of a plant's life cycle being inextricably linked to the seasonal rhythms of its specific environment. It explains why a plant from a temperate climate requires a simulated winter, and why a purely tropical plant would not. Finally, after what felt like an eternity, I removed the container from the refrigerator and placed it in a warm, sunny location. Within a month, tiny sprouts began to emerge from the peat and sand mixture. The first leaves were not the elegant, tubular pitchers I had so eagerly awaited. They were small, flat, and non-carnivorous, a testament to the plant's juvenile stage. This first-year growth was slow, with the seedlings only reaching a height of 1 to 2 inches by the end of their first season. This period of juvenility is a lengthy one for Sarracenia, with many species taking 3 to 6 years before they are mature enough to form their first pitchers or flowers. The process underscored the deep-seated patience required for this type of cultivation. Year 2 and 3, the patient wait and the tropical sprout. As the Sarracenia seedlings dutifully grew, slowly but surely producing new leaves from their rhizomes, the tropical Nepenthes seeds offered a different kind of lesson. One in immediate gratification. I had planted them at the same time, but without the need for cold stratification. They had already begun their journey. Their tendrils, extensions of the leaf midrib, were starting to develop small, spherical buds at their tips. Day by day, these buds would swell, changing in color and shape, and then, a small frill would unfurl, revealing the delicate, new pitcher within. The process of a single pitcher growing from a bud to a full-sized trap is a captivating display of natural engineering that takes approximately 1 month. It is a highly sensitive process, and fluctuations in light, temperature, or humidity can cause the bud to fail and turn black, never to open. This is why, in the early stages, observing new leaf and tendril production is often a better indicator of a plant's health than the success of its pitches. This visible, dynamic growth provided a stark contrast to the slow, subterranean progress of the Sarracenia and highlighted a fundamental difference in their evolutionary strategies. As the Nepenthes grew, it began to exhibit a fascinating biological phenomenon known as leaf dimorphism. The plant started producing 2 distinct types of pitchers. The first were large, lower traps that sat on the ground, often richly colored and ovoid in shape. As the stem of the plant matured and began to climb, it produced a second kind, the upper, or aerial, pitchers. These were usually smaller, differently colored, and had a loop in the tendril, which the plant used to wrap around nearby supports, a crucial adaptation for its vining growth habit. The disparate life cycles of the 2 genera, one requiring a long, dormant period and a patient wait for maturity, and the other designed for constant, climbing growth, are not random variations. They are finely tuned responses to the unique ecological niches. The Sarracenia is a plant of the open bog, where its life cycle must align with the dramatic seasonal changes of a temperate climate. Its entire survival strategy revolves around storing energy in a rhizome to endure the cold, non-growing season, a process that is non-negotiable for its long-term health in cultivation. The Nepenthes, by contrast, is a plant of the dense, tropical jungle. Its vining habit is a strategy to compete for and reach the full sunlight of the canopy, where its flowers and upper pitches can thrive. This understanding of their evolutionary drivers directly informs their cultivation needs. A Sarracenia thrives in a full sun, open environment that mimics its bog habitat, while a Nepenthes needs the high humidity and bright, indirect light of a greenhouse or terrarium, which simulates the dappled light and moist air of its jungle home. This difference in their ecological histories explains why there is no single, universal approach to growing pitcher plants. Success depends entirely on providing a physical and climatic environment that honors the unique evolutionary path. Year 4, the digestive cocktail and the perilous trap. By year 4, both my Sarracenia and Nepenthes had reached a stage of robust maturity. The The Sarracenia pitchers, now reaching their full size, were ruthlessly efficient, often filling to the brim with flies, wasps, and ants. The Nepenthes had claimed its own victims, and I found myself studying the anatomy of the trap with a new level of fascination. It begged a profound question. How, without a mouth or stomach, does a plant eat its prey? The answer, I learned, lay in a masterful combination of ingenious physical design and sophisticated biochemistry. The pitcher itself is a perfectly engineered pitfall trap. The journey begins with the plant's lure, a combination of vibrant coloration, sugary nectar, and sweet scents that attract insects. Nectar glands are often found on the underside of the lid, or operculum, which also serves to keep rainwater from diluting the digestive fluid inside. The insect, in its pursuit of the nectar, is forced to walk along the rim, or peristom, which is extremely slippery when wet due to its waxy surface. A single misstep sends the insect tumbling down into the deep, tubular cup. The inner walls are coated with a waxy substance and lined with downward-pointing hairs, creating an inescapable labyrinth from which the victim cannot climb out. At the bottom of this perilous trap lies a pool of digestive fluid. Far from being a simple puddle of water, this fluid is a potent, acidic cocktail of hydrolytic enzymes produced by glands on the inner surface of the pitcher. This fluid is highly acidic, often reaching a pH of 2 to 3, a level comparable to the gastric acid in the stomachs of insect-eating animals and the human stomach. This acidity, primarily generated by inorganic acids like hydrochloric acid, serves a dual purpose. It acts as a barrier against foodborne pathogens and creates the optimal environment for the digestive enzymes to function. The range of enzymes identified in Nepenthes pitcher fluid reveals a highly specialized and comprehensive digestive system. The primary enzymes for breaking down the proteins of captured insects are the aspartic proteases, a group that includes Nepenthesin 1 and 2, isolated from species such as N. alata, N. distillatoria, and N. gratulus. These enzymes are critical for degrading the soft tissue of the prey. To break down the tough outer shell of an insect, the plant secretes chitinases, which degrade the chitin found in arthropod exoskeletons. Further digestion is managed by lipases and esterases, which break down fats, and various other enzymes such as phosphatases, peroxidases, and glucanases, which collectively ensure a full breakdown of the prey. The existence of this complex enzymatic repertoire reveals that the pitcher is a highly evolved, all-in-one organ, unlike an animal's digestive tract, where food travels through a series of specialized organs. The pitcher leaf performs every function, luring, trapping, digestion, and nutrient absorption, in a single place. This is a targeted and efficient biological process, but it is not without its limitations. The plant's digestive machinery is finely tuned to its natural diet of insects. This is why a common practice among novice growers, feeding the plant raw meat, can be so detrimental. The plant's digestive fluid lacks the specific enzymes necessary to break down the high fat content in animal meat, which can cause the pitcher to rot and become a site for harmful microbial growth. Understanding this biochemical specialization is a critical step in moving from a casual grower to a true steward of these remarkable plants. Year 5 and beyond. Symbiosis and the full cycle. The journey from a seed to a fully mature plant culminates in a final, breathtaking stage of the life cycle. For my Sarracenia, this was the year it produced its first flowers, a surreal sight of bright yellow petals unfolding on a long stalk. For the Nepenthes, it was the year its upper pitchers grew to an astonishing size, displaying not only their carnivorous efficiency but also their capacity for more complex relationships. I had learned that these plants were not just passive traps, they were participants in a web of sophisticated, and sometimes bizarre, symbiotic relationships. The most extraordinary example of this is the mutualism observed in certain high-altitude Nepenthes species, such as N. Louis, N. raja, and N. macrophylla. While most Nepenthes are masters of trapping insects, These species have developed an alternative nutrient gathering strategy. They have modified the pitchers to function as toilets for mountain tree shrews and summit rats. The pitchers have large orifices and lids that are reflexed to approximately 90 degrees, creating a shape that forces the mammal to sit astride the pitcher to feed on a sugary exudate produced on the lid's inner surface. While the mammal feeds, it defecates directly into the pitcher. The plant receives a valuable source of nitrogen and phosphorus from the feces, which in some species can account for up to 100% of their full air nitrogen content. This is an incredibly specialized and mutually beneficial arrangement, where the plant provides a consistent food source in a nutrient-scarce environment, and the animal provides the plant with essential fertilizer. This intricate, toilet pitcher strategy stands in stark contrast to the digestive process of Sarracenia and other genera like Heliophora, which rely on a different kind of symbiosis. While Nepenthes produces a powerful, self-contained cocktail of digestive enzymes, Sarracenia has a fluid that is little different from pure water. Instead of producing its own enzymes, the plant hosts a diverse community of organisms, called inquilines, within its pitcher. This miniature ecosystem, or phytotelm, includes a vast array of bacteria, protozoa, fungi, and even mosquito larvae. It is this microbial community that performs the bulk of the digestive work. These inquilines secrete a range of enzymes, including chitinases and proteases, that break down the captured prey, making the nutrients available for the plant to absorb. The plant, therefore, is not a self-sufficient factory but a landlord for a symbiotic community that provides essential digestive services. Finally, as the year drew to a close, my Sarracenia underwent its most dramatic transformation. The vibrant, upright pitchers began to turn brown and wither, a natural process that would continue until the entire rosette of leaves died back. The plant was entering its winter dormancy, a crucial period of rest that is indispensable for its long-term health. During this time, the plant relies on its underground rhizome to survive, and it can withstand freezing temperatures, with snow and dead foliage acting as a natural insulator. This dormant state is a clear demonstration of the plant's deep-seated adaptation to a seasonal climate, and it is a rhythm that must be respected in cultivation to ensure its survival for years to come. Conservation, the fragile king and queen. As I witnessed my collection flourish, it was impossible not to reflect on the precarious state of these same plants in the wild. The very qualities that make them so attractive to gardeners, their beauty, rarity, and unique biology, are also what make them vulnerable. The primary threats facing both Nepenthes and Sarracenia species are deeply connected to human activity. A major factor is the rapid destruction of the unique wetland habitats due to agriculture, human development, and logging. These plants have evolved over millennia to thrive in very specific, nutrient-poor environments, and the widespread alteration of these ecosystems leaves them with nowhere to go. In addition, the illegal collection and poaching of living plants and seeds for the rare plant trade is a significant threat. Their beauty and rarity create a high demand among collectors, which incentivizes the illegal harvesting of wild specimens. These threats have had a devastating impact on many species. On the IUCN Red List of Threatened Species, 35 species of the genus Nepenthes are listed as vulnerable or endangered, and 10 species are classified as critically endangered. Among the Sarracenia, the green pitcher plant, Sarracenia aureophila, is considered the most endangered, with its range limited to a handful of sites in the southeastern United States. It is listed as critically endangered by the IUCN and is protected under the U.S. Endangered Species Act. The whitetop pitcher plant, Sarracenia leucophila, is also listed as a vulnerable species. In response to these threats, international and national conservation measures have been put in place. Species such as S. aureophila and N. raja are listed on Appendix I of the Convention on International Trade in Endangered Species, CITES, which prohibits all commercial international trade in wild-sourced specimens. This shift in legal protection forces the market to rely on a sustainable, ethical source of plants. It is within this context that the role of the hobbyist becomes a critical part of the solution. By purchasing only nursery-propagated plants from reputable sellers, a gardener can help reduce the demand for wild-collected specimens and, in a sense, help fund the ex situ conservation efforts underway in botanical gardens and seed banks. The very act of growing these plants, when done ethically, can be an act of preservation. Epilogue and the Comprehensive Care Guide. The 5-year journey of my pitcher plants has been a lesson in more than just cultivation. It has been an education in the intricate, interconnected, and often surprising ways that life adapts to survive. From the patience required to honor a plant's seasonal cycle to the sheer wonder of its complex biochemical machinery and its hidden symbiotic relationships. The experience has been a testament to the fact that there is always more to learn. For those who wish to embark on their own journey, a thorough understanding of the care requirements for these unique plants is essential. Light, soil, and water requirements. Both Sarracenia and Nepenthes have specific and distinct needs that must be met for them to thrive. Sarracenia prefers full, direct sun for at least 6 hours daily. A lack of light will result in weak, poorly colored pitchers. In their natural habitat, they are bog plants, but they are very sensitive to dissolved salts and minerals. As such, they must be watered with distilled water, rainwater, or reverse osmosis water. During the growing season, they prefer to be kept in a tray with about 2 centimeters of water. A method often described as having wet feet, but dry ankles, as the rhizome should remain above the waterline in taller pots. The recommended soil mix for Sarracenia is a combination of sphagnum peat moss mixed with lime-free horticultural sand or perlite in a 2-to-1 ratio, which provides the moist but well-draining conditions they require. Regular potting soil should be avoided at all costs, as it retains too much moisture and can harm the plants. Nepenthes, by contrast, thrives in bright, indirect light. In their native habitat, they often grow in dappled light or on mountain ridges that experience frequent fog. And too much direct sun can severely burn them. They have no tolerance for waterlogged soil and should never be left standing in a tray of water. Instead, they should be top-watered with distilled, deionized, or rainwater, allowing the medium to become moist but not soaked. Their preferred soil mix is looser and airier than Sarracenia's, typically a ratio of 1 part orchid bark, 1 part perlite, and 1 part chopped sphagnum moss. Feeding and fertilization. Pitcher plants are adapted to nutrient-poor environments, and they derive most of their nutritional needs from the insects they capture. When grown outdoors in a suitable climate, they are fully capable of catching more than enough prey on their own. For indoor cultivation, where insects may be scarce, it is possible to hand-feed the plants with dried insects, such as dried crickets or bloodworms, or with specialized fish food. Alternatively, a small pellet of Osmocote fertilizer can be placed into a single pitcher once a month. Or a diluted orchid fertilizer can be applied as a foliar spray. It is crucial to never use regular chemical fertilizers, which contain high levels of salts that can burn and kill the plants. Likewise, avoid feeding them raw meat or other human food, as their digestive systems are not equipped to break down fat, which can lead to rot. Dormancy and pruning. One of the most critical aspects of cultivation is respecting the plant's natural life cycle. Sarracenia species require a mandatory winter dormancy period of 2 to 3 months, during which watering should be reduced. The plants should be kept in a cooler location, ideally between 4 degrees Celsius and 10 degrees Celsius, and will die back to their rhizome. This rest period is essential for their long-term health and vigor. The dead leaves and pitchers should be left on the plant throughout the winter to provide insulation and should only be trimmed off in the spring. In contrast, Nepenthes species do not experience a dormancy period and require warm temperatures and consistent care year-round. While they do not go dormant, they will naturally shed their older pitchers and leaves as they age or if the plant is stressed. These can be trimmed off once they are entirely brown or black. Pests and diseases. For healthy, well-cared-for plants, pests and diseases are generally negligible. However, there are a few common issues to be aware of. Pitcher rot can occur if a pitcher accumulates too many insects, which can lead to a mushy, brown discoloration. This does not harm the rhizome and can be managed by simply cutting off the affected pitcher. In some cases, wasps may chew holes in the pitchers, causing them to collapse, but this does not cause long-term harm to the plant. For container-grown plants, scale insects can occasionally be a problem, particularly during dormancy. Good ventilation is important for both genera to prevent fungal infections. This comprehensive care guide serves as a summary of the most important lessons learned over the journey. It reinforces the understanding that these are not just ornamental plants but living beings with unique, deeply ingrained needs. By approaching their care with a sense of stewardship, a gardener can ensure the long-term health of their collection while contributing to the preservation of these remarkable botanical wonders.